Welcome back to the deep dive. Today we are wrestling with a technology that it feels increasingly like science fiction becoming reality. CRISPR-Cas9. We've known for years, obviously, that the system lets us edit DNA. But the cutting edge now, it's really about precision. It's about doing these surgical, really nuanced edits to solve problems we thought were, well, maybe impossible just a few years back.
Right.
And that huge power, of course, brings complexity, technical complexity, and some really profound moral questions, too.
It absolutely does. And our mission today is focus on how that technical power is being used for one of the toughest challenges in genomics, dominant genetic diseases. So we're going to dive into something called allele-specific editing. But at the same time, we really have to look at the ethical roadmap that, the international community is trying to draft for all this. It's urgent.
Yeah, and that technical challenge is really key to understanding the breakthrough here. So with a dominant genetic disorder, you've got two copies of a gene, right? One's normal, the wild type, and the other one's defective. And the problem isn't just that the defective one doesn't work. Often, the protein it makes actively messes things up. It interferes with the normal protein.
Exactly. That's the interference we call the dominant negative mechanism. And it means for a treatment, you can't just knock out the whole gene. Because you'd silence the good functioning copy too.
So you lose the benefit.
You lose the benefit. The real trick, the genomic surgery, is silencing only the mutant allele, the bad one, while making sure that good normal allele is left completely untouched.
Okay, so that's the goal. Where are we seeing researchers actually achieve this level of precision? Let's maybe start with a case study.
Yeah, a good place to start is Leber congenital amaurosis, or LCA7. It's a dominant eye disease linked to the CRX gene, causes serious problems with photoreceptors in the retina.
Right.
So to test out gene editing ideas, researchers use these things called LCA7 retinal organoids. Basically, mini retinas grown in a dish carrying the disease mutation.
Like a model system.
Exactly, a model system. And what did they do? They applied allele-specific CRISPR-Cas9 editing, designed very carefully to knock out only the mutant CRX allele.
And the results in these mini retinas.
They saw what they described as a moderate rescue of the photoreceptor problems. Now, moderate might not sound amazing, but it's huge proof of concept.
Right. It shows it's possible.
It shows selective silencing is possible for these dominant diseases like LCA7. And, that opens the door for trying this in other similar conditions.
That's fantastic progress, especially when you can target one specific mutation like that. But here's where genetics often gets complicated, right? The sheer variety. Let's maybe look at another example. Charcot-Marie-Tooth disease type 2E, or CMT2E. It's a type of genetic neuropathy, nerve damage, caused by mutations in a gene called NEFL.
Yeah, CMT2E is another classic dominant negative situation. The faulty protein, it's called neurofilament light chain protein, messes up the structure inside nerve axons. Really critical stuff. But there's a bit of good news here, biologically speaking. The normal NEFL gene is what we call haplosufficient.
Meaning one good copy is enough.
Exactly. If you have just one functioning copy, you're generally fine. So theoretically, if you could just knock out the bad copy, the dominant mutant one, it should be safe and well tolerated by the patient.
Okay, that makes sense. But you mentioned variety. What's the core problem for treating CMT2E with this allele-specific approach?
The problem is scale. CMT2E isn't caused by just one mutation. It can be caused by over 50 different missense mutations scattered all across the NEFL gene.
Wow, 50.
50. So imagine trying to develop a specific CRISPR therapy for each one. 50 different drugs, 50 different clinical trials. The researchers themselves called it laborious and impractical.
Yeah, you can see why. You need a different strategy, something that doesn't care which of those 50 mutations someone has.
Precisely. And that need led directly to this really innovative strategy. Haplotype editing.
Haplotype editing. Okay, break that down.
So instead of targeting the disease mutation itself, which could be any one of those 50, they target something else that's physically linked to it on the chromosome.
Linked?
Yeah, they go after common non-coding variations in the DNA, things called single nucleotide polymorphisms, or SNPs, tiny changes that usually don't do anything themselves.
Wait, so let me get this straight. They're targeting these little harmless DNA variations, basically genetic markers that just happen to be inherited together with the bad gene copy because they're physically close on the chromosome.
That's exactly it. They're inherited in cis on the same chromosome. The mutation itself becomes almost irrelevant to the targeting strategy.
Okay, so how does targeting the marker help get rid of the bad gene?
They use paired guide RNAs, gRNAs. One targets an SNP on one side of the gene, the other targets an SNP on the other side.
Like bookends.
Like bookends. When the Cas9 enzyme cuts at both of those SNP sites, the cell's repair machinery often just deletes or excises the entire chunk of DNA in between, including the entire mutant gene sequence.
Wow. So you just cut out the whole faulty copy, markers and all, without ever needing to know the specific mutation inside it. That's clever.
It's very clever, and it makes the approach mutation agnostic.
And is this actually practical? Like, do enough people have these targetable SNP markers near the gene?
That's the crucial question, right? And the population data looks really promising. Researchers looked at the 1,000 Genomes Project data and found that somewhere between 32 and 41% of people are heterozygous, meaning they have different SNPs on their two chromosomes for pairs that could be targeted this way around the NEFL gene.
So potentially a third to almost half the population could benefit from this kind of approach for this gene alone. That's huge.
It completely changes the drug development picture. Remember the 50-plus mutations for CMT2E?
Yeah, the impractical problem.
Well, with this haplotype editing strategy, the researchers showed that using just 8 different combinations of Cas9 and guide RNAs could potentially treat over 50% of patients.
From 50 down to 8.
And it gets better. If they added just one more universal guide RNA, one that works on almost everyone, they could use a total of 9 combinations to potentially treat over 90% of the patient population.
Wow. 9 therapies to cover 90%. That basically solves the complexity and the scaling problem in one go.
It really does. It's a potential pathway to making these therapies feasible.
Okay, that's a massive leap forward in terms of efficiency and potentially accessibility. But biology is never quite that simple. The research apparently uncovered a kind of surprising technical challenge with using those paired guide RNAs, the inversion surprise.
Yes, exactly. So when you make those 2 cuts with the paired gRNAs, you want the cell to just delete the bit in between that's excision.
Right, get rid of the bad copy.
But cells sometimes repair that double break differently. Sometimes, instead of deleting the segment, they flip it around, invert it, and stitch it back in.
Okay, an inversion. But wouldn't flipping the gene also break it? Like scramble the code?
You'd think so. But that was the surprise. The inversion often failed to reliably shut down the mutant gene's expression. The faulty protein was still being made.
Why wouldn't flipping it stop it from working?
Well, the analysis showed that the inversion often didn't actually separate the main coding part of the gene from its essential control elements, the promoter, the regulatory sequences that tell the cell to read the gene.
So even though it was backwards, the on switch was still connected to the message.
Pretty much. So the cell could still transcribe the mutant gene, even inverted, which meant the therapy wasn't working reliably if an inversion happened. For it to work consistently, you really needed the excision, the deletion.
Okay, so the goal then became, how do we maximize the chance of getting that therapeutic excision, or at least make sure that even if an inversion does happen, the bad gene still gets silenced?
Right. And they came up with 2 really smart solutions. First, they tweaked the guide RNA pairing strategy. They called it biallelic gRNA pairing. Yeah, meaning it targets something present on both alleles, mutant and wild type. They kept their allele-specific SNP guide RNA, the one targeting the marker near the bad copy.
Okay.
But they paired it with a new universal guide RNA that targeted a sequence inside the gene itself, specifically in the first intron, which is a non-coding bit near the start.
So one cut is allele-specific outside the gene, the other is universal inside the gene.
Exactly. So now, even if the DNA segment inverts instead of deleting, that internal cut point is guaranteed to disrupt the gene's structure right near the transcription start site.
So the inversion itself becomes disruptive. Clever. It's like building in a failsafe.
It's planning for the failure mode, exactly. It ensures the mutant allele gets inactivated, whether it's excised or inverted.
That's really elegant, making sure the break happens somewhere useful regardless. Okay, what was the second solution? You said it was simpler.
Almost amusingly simple, yeah. It was just a physical boost. They found that adding a tiny little piece of DNA, a 60-nucleotide single-stranded oligonucleotide donor, or SODN, along with the CRISPR machinery, it just somehow encouraged the cell's repair pathways to favor excision. It increased the desired excision frequency by like 30 to 50% in their experiments.
Just adding a little piece of DNA nudges the cell towards deletion.
Seems like it. Sometimes biology just needs a little physical encouragement,
Okay, so they optimize the system to favor deletion and make inversions therapeutic too. But with all this cutting and pasting, we have to come back to specificity.
Yeah.
How sure were they that they were only hitting the bad allele and not the good one?
Critically important question. And the initial results with just the allele-specific SNP pairs were actually fantastic. They were achieving like 92 to 99% specificity, really good at distinguishing the target allele.
That's very precise.
Very precise. However, when they introduced that universal biallelic gRNA for the fail-safe mechanism, things got a bit more complicated. Some of those pairings showed decreased specificity, sometimes down to like 64 to 77%.
Oof, that's a significant drop. Hitting the good copy potentially a quarter of the time or more.
No. Exactly. It really highlights that every single guide RNA combination has to be rigorously tested for off-target effects, especially allele specificity. They actually had to use some pretty novel digital PCR methods just to measure it accurately. It confirms that specificity, making sure you only edit what you intend to edit, remains a major bottleneck, or at least a critical checkpoint.
Absolutely. But despite that challenge, the overall biological outcome in their CMT2E models was positive, right? Did the approach actually rescue the cellular problems?
Yes, the functional rescue was clear. When they successfully excised the mutant NEFL allele using these optimized strategies, they saw normalization of the nerve cell pathology. That problematic buildup of the NfL protein in the cell bodies decreased.
That's good.
And the levels of NfL in the neurites, the nerve projections where it's needed, were restored. So bottom line, the mechanism works in the model system, and the haplotype strategy makes it potentially scalable.
Okay, so the technical ability is really advancing incredibly fast.
It is, and that technical success, that ability to solve the complexity riddle, treat 90% of patients with just 9 combinations, forces us, really, to confront the huge ethical implications of having this kind of power. How is it going to be used?
Let's maybe start with the less controversial stuff. Current applications that are non-heritable somatic therapies. We're already seeing gene knockout being used, for instance, in cancer immunotherapy.
Absolutely. Things like improving CAR T-cell therapy, making those engineered immune cells safer or more effective by knocking out certain genes. Those changes aren't passed on. It's just treating the individual patient. That's happening now.
And beyond human therapy, there are massive applications in other areas, right? Things with potentially global impact.
Oh, definitely. Look at food security. CRISPR is being used or proposed to make crops more nutritious, more resistant to drought or pests, maybe improve livestock. You mentioned nearly 800 million people worldwide are undernourished. This technology could directly address that.
Huge potential benefit there. What about something like gene drives? That sounds a bit more sci-fi.
Gene drives are fascinating and slightly scary. The idea is to use CRISPR to make a genetic change spread incredibly quickly through a wild population.
Oh.
Basically, you engineer the gene edit so it copies itself onto the other chromosome during reproduction, so nearly all offspring inherit it instead of just half. It lets you rapidly change an entire species in a local environment.
And the application there would be disease control?
Primarily, yes. The main example is engineering mosquitoes so they can't carry malaria or dengue fever, and then driving that trait through the mosquito population to wipe out the disease transmission.
Incredible potential for public health. But I can see the risks, too. You're altering an entire ecosystem. What if it goes wrong?
Exactly. The potential for unintended consequences is massive. That's why there's so much focus on building in safety features like off switches or making the drive geographically limited. It's powerful, but needs extreme caution.
Okay, so that's non-human uses. When we swing back to humans, the ethical lines get blurry very fast, don't they? Particularly around enhancement versus therapy.
This is a huge gray area. Where does treating a disease end and enhancing a human begin?
Right. I mean, should we use CRISPR to give someone better vision than 20/20? Or improve memory? Or make someone taller?
It's a slippery slope, potentially. If we edit a gene to fix, say, dangerously high cholesterol, everyone agrees that's therapy.
Sure.
But what if we use gene editing to boost someone's natural athletic ability? Or maybe even cognitive function? Is that still therapy? The source material really highlights how difficult and maybe dangerous it is to draw that line definitively.
And that leads us right into the deepest ethical water. Editing the human germline. Making changes that are heritable, passed down through generations.
Yes, germline editing. That's the core controversy, because you're not just affecting one person, you're potentially altering the human gene pool.
What's the sort of official stance on that right now? Are there guidelines?
Well, major bodies like the U.S. National Academies of Sciences, Engineering, and Medicine, NASM, have weighed in. Their position is generally supportive of somatic editing, the non-heritable kind. For germline editing, they're much more cautious. They've suggested it might be permissible someday, but only under incredibly strict conditions. For serious diseases with no other treatment options, with robust oversight and, crucially, long-term follow-up of the children and future generations.
They're the very high bar, and maybe not even then.
A very, very high bar. And even if the technical hurdles are cleared and the ethical guidelines allow it for specific diseases, there's another huge ethical issue: access.
The cost.
The cost. These therapies, especially early on, are likely to be incredibly expensive. Who gets them? Does this create a new kind of genetic divide between the rich and the poor?
Yeah, that raises serious questions about fairness, about distributive justice, especially when you think, as you mentioned, a lot of the basic CRISPR research was funded by public money, by taxpayers.
Exactly. If the public paid for the foundation, should the resulting cures be priced out of reach for most people? It's a major concern.
And hovering over this entire discussion, especially germline editing, is the shadow of, well, eugenics.
Yes. It's impossible and irresponsible to discuss this without acknowledging that history. There's a deep, legitimate fear that allowing germline editing, even with the best intentions for medical therapy, could open the door to non-medical enhancement and eventually revive the idea of improving the human race through selective genetics.
We have to remember the history there. Things like the Buck v. Bell Supreme Court case in the U.S., which upheld a forced sterilization based on flawed ideas about heredity.
And tragically influenced Nazi eugenics programs later on. That historical context demands extreme caution and vigilance against any potential misuse, even if it starts with purely therapeutic goals.
So pulling it all together, the technical side is just rocketing forward. We've gone from basic gene knockout to this incredibly sophisticated allele-specific, mutation-agnostic haplotype editing. We're showing we can potentially tackle diseases like CMT2E that seemed untouchable before.
Absolutely. The technical progress is undeniable. Innovations like solving that inversion problem using an intragenic cut show how rapidly the field is refining these tools. We can do things now, or are very close to doing things, that were firmly in the realm of science fiction just a decade ago.
But that speed is the challenge, isn't it? The science, the technical capability, seems to be constantly outrunning our ethical frameworks or regulations, our societal consensus on how to use it.
It really does feel that way. We have the tools now to make these complex, potentially permanent changes, not just to individuals, but possibly to the human germline itself.
Which brings us to the final thought for you, our listener. The question isn't just, can we perform these incredibly complex edits? The research, like the haplotype editing for CMT2E, suggests we're getting very close to a yes on that. The deeper question is, should we? Given the unknowns about long-term effects, the difficulties in drawing the line between therapy and enhancement, and the undeniable historical shadow of eugenics, should the global community maybe start thinking about new or revised international rules? Perhaps something modeled on agreements that govern other powerful, potentially dual-use technologies like the Geneva Conventions or the UN Conventions on Biological Weapons, to formally acknowledge and regulate the profound power of genomic engineering.
It's a heavy question, but one we absolutely need to be asking now. As the technology continues its rapid advance, we really encourage you to dig into the source material yourself and keep grappling with these essential issues.
Definitely food for thought. Until next time on The Deep Dive, keep digging deeper.